Hello everyone, I am back. I am Dr. Deepika Malik. Welcome to the fourth session of Downstream Processing. In the first session, we have discussed cell disruption methods. In session 2, we started with the stage 1, that is solid-liquid separation, and discussed about filtration methods in detail. The session 3 was in continuation with solid-liquid separation only, and there we discussed centrifugation, sedimentation and flocculation. Now in our fourth session, we'll discuss our stage 2, that is concentration methods. The filtrate that is free from suspended solids like cells or cell debris usually contains 80 to 98% of water. The desired product in it is very minor constituent. Therefore, the water has to be removed to achieve the product concentration. The commonly used techniques for concentrating biological products are liquid-liquid extraction, Aqueous Two-Phase Extraction, Precipitation, Evaporation for solvent recovery using distillation plant that can be operated in batch as well as continuous form, and then Supercritical fluid extraction. Now let us discuss these steps one by one in detail. Coming to the first part, that is liquid-liquid extraction. The separation of a component from a liquid mixture by treatment with a solvent in which the desired component is preferentially soluble is known as liquid-liquid extraction. For a better understanding, let's see the diagram. Here the feed or broth containing water is denoted by A and the solute or desired product is denoted by B, as you can see in the first part of the diagram. In next step, it is mixed with a new solvent denoted by C and which is red in color. Since water and the solvent do not mix with each other, therefore two separate phases A and C are obtained after shaking. The solute partially passes to the solvent C, leading to its extraction. Feed which gets deficient in solute is termed as raffinate and the new solvent rich in solute is termed as the extract. Now this extract, which is basically solvent rich in our desired product or the solute, can further be treated or processed by gravitation or other separation technique like centrifugation so that the solvent can be recovered back, whereas our desired product gets concentrated. Here we are using organic solvent for the separation, therefore this technique is also termed as solvent extraction. Next method is Aqueous Two-Phase Extraction method. Although liquid-liquid extraction is a well-established technology in chemical processing, but the use of organic solvents has limited application in the processing of sensitive biologicals. Therefore, for the extraction of biologically active polymers such as proteins, enzymes and nucleic acids, it is necessary to have a two-phase extraction system that will not denature the biopolymers. This aqueous two-phase system have a high water content solvent which offers low interfacial surface tension and are regarded as being biocompatible, meaning they are more compatible with living organisms and biomolecules. The phase separation in aqueous two-phase system happens when hydrophilic polymers are added to an aqueous solution and their concentration reach a certain threshold. Therefore, at this point, the mixture forms two distinct liquid phases that do not mix with each other, that is, they are immiscible. These phases are both composed of water, but they have different composition and properties. These two distinct liquid phases can be prepared by mixing a polymer such as polyethylene glycol and a salt solution like ammonium sulfate, or by adding two different polymers. Now the important points to note for this technique is that water is the main component in the two phases, but the two phases are not miscible. Cells and other solids remain in one phase while the proteins are transferred to the other phase. The distribution of the desired product is based on its surface and ionic character and the nature of phases, and the separation takes much longer time by this method as compared to liquid-liquid extraction. Now to make you understand this process better, let's see the diagram on the next page. This process of extraction basically involves two steps. The first step is mixing of sample, that is the fermentation broth, with the two-phase solvent, that is your polyethylene glycol and salt mixture for extraction. And then the separation of phases is carried out. So as you can see in the diagram, we have a sample that includes cell debris, biopolymers and proteins. When this sample is mixed with two-phase aqueous system containing polyethylene glycol and salt solution, it is observed that cell debris will settle in the denser or the bottom phase of the tube. The top or the lighter phase contains biopolymers and proteins which are shifted to yet another tube containing a fresh salt-rich bottom. After centrifugation is carried out, we can see the biopolymers now settle at the bottom and the proteins remain suspended in the upper lighter phase. The proteins are further shifted to another tube containing again fresh salt solution, and after centrifugation, these target proteins settle at the bottom of the tubes which can further be recovered. The polyethylene glycol phase at the top can further be recycled. Finally, ultrafiltration is taken up for the final recovery of the desired product. The next slide shows a comparison between the liquid-liquid extraction and aqueous two-phase extraction system. First parameter of comparison is the principle. Liquid-liquid extraction involves the transfer of a solute, that is our target compound, from one immiscible liquid phase, that is an organic solvent, to another immiscible liquid phase, which can be an aqueous or organic phase. The transfer is based on the solute's differential solubility between the two phases. Whereas aqueous two-phase extraction involves the formation of two immiscible aqueous liquid phases using phase-forming agents such as polymers or salts. The target compound partition between two phases based on their affinity for each other. Liquid-liquid extraction typically utilizes an organic solvent as one of the liquid phases, which can dissolve the target compound selectively, whereas both liquid phases in aqueous two-phase extraction are aqueous, which can be advantageous when working with biological compounds since it avoids the use of organic solvents that may be toxic or harmful to biomolecules. Liquid-liquid extraction can achieve selectivity just by choosing solvents that preferentially extract the target compound while leaving impurities behind. On the other hand, aqueous two-phase extraction allows for fine-tuning of partitioning and selectivity by adjusting the composition and properties of the two liquid phases. Parameters such as pH, temperature, salt concentration, and polymer type can be modified to enhance the partitioning of the target compounds. Finally, the liquid-liquid extraction can be challenging to scale up to large volumes due to the need for efficient phase separation and the potential formation of emulsions during extraction. Now, these emulsion formation can actually complicate the separation process and reduce overall efficiency. So, these emulsions basically delay the separation of two liquids. So, just to brush up, an emulsion is a mixture of two or more liquids that are usually immiscible, but under specific conditions, they'll manage to form a homogeneous mixture. On the other hand, aqueous two-phase extraction is readily scalable and can be adapted for both small and large-scale operations. It can be performed in batch or continuous modes, making it versatile for different production scales and requirements. Next method is precipitation method. Precipitation plays a crucial role in the concentration of biological products during the downstreaming process. It may be conducted at various stages of the product recovery process. The mechanism of precipitation is that soluble compounds in solution become insoluble due to different chemical reaction parameters. By adjusting the pH, temperature, and concentration of the precipitating agents, the solubility of the target molecule can be altered. This results in a reduction in its solubility, leading to the formation of solid precipitates. As you can see in the diagram, the precipitating agent is added to a solution, resulting in an insoluble complex formation, which further leads to precipitation of the desired or target product. These precipitates can be separated from the liquid phase through the techniques such as filtration, centrifugation, or sedimentation that we have already discussed. The recovered solid can then further be processed to obtain the purified biological product. Depending on the downstream processing requirement, the precipitated biomolecules may need to be resolubilized in a suitable solvent or buffer solution. This step allows for the recovery of the concentrated product in a more usable form for subsequent processing or formulation. It is possible to obtain some products or to remove certain impurities directly from the broth by precipitation, or use the technique after a crude cell lysate has been removed. The efficiency and selectivity of the precipitation can vary depending upon the characteristics of the biological product and the precipitating agent used. The next slide shows different precipitating agents that can be used to make our target compound insoluble. First is the use of acids or bases to change the pH of a solution until the isoelectronic point of the compound is reached. Here Pi denotes isoelectronic point, where there is no overall charge on the molecule and the solubility of the target compound is decreased. Salts such as ammonium and sodium sulfate are used for the recovery and fractionation of proteins. The salt removes water from the surface of the protein, revealing hydrophobic patches which come together, causing the protein to precipitate. The most hydrophobic proteins will precipitate first, thus allowing fractionation to take place. Organic solvents such as methanol can be used to precipitate dextrins out of the broth. Chilled ethanol and acetone can be used in the precipitation of proteins, mainly due to changes in the dielectric properties. Non-ionic polymers such as polyethylene glycol can be used in the precipitation of proteins and are similar in behavior to organic solvents. Polyelectrolytes can be used in the precipitation of a range of compounds in addition to their use in cell aggregation. Protein binding dyes such as triazine bind to and precipitate certain classes of proteins. Affinity precipitants are an area of much current interest in that they are able to and precipitate compounds selectively. Next we have is evaporation for solvent recovery using distillation plants. The solvents used for extraction are in excess, therefore they need to be recovered to reduce cost. Evaporation is commonly used in solvent recovery processes, and one method that is frequently employed is through a distillation plant. Distillation is a separation technique that utilizes the difference in boiling points of components in a liquid mixture to separate them. In this, components are separated from a mixture via continuous heating and cooling. Distillation may be achieved in 3 stages. First is evaporation, that is the removal of solvent as a vapor from a solution. Second stage is the vapor-liquid separation in a column to separate the lower boiling more volatile component from the less volatile components. Third stage is the condensation of the vapor, that is to recover more volatile solvent fractions. Now there are 2 major forms of distillation. First is batch distillation and second is continuous distillation. Batch distillation is a method of separating components in a mixture batch-wise, as the name suggests. The batch distillation is easy to be carried out. Several different components can be separated using a single distillation column. In this method, the separation via distillation is done repeatedly. In batch distillation, the vapor from the boiler passes up the column and is condensed. Part of the condensate will be returned as a reflux for counter-current contact with the rising vapors in the column. The distillation is confined until a satisfactory recovery of the lower boiling or more volatile components have been accomplished. It is labor-intensive as it requires manual intervention for charging the apparatus, monitoring the process, and collecting different fractions. Batch distillation is also time-consuming as compared to continuous distillation. The process involves heating the mixture, waiting for separation, and collecting fractions before the next batch can be started. The design of the equipment is also simpler as compared to continuous distillation system. So the important thing to note here in the diagram is that we have one condenser which is carrying out the separation and helping in collecting different fractions. Whereas continuous distillation is the method of separating components in a mixture using a continuous process. There are no interruptions for the process until the completion of the distillation. Continuous distillation is generally more efficient than batch distillation for large-scale operations. It allows for a higher throughput, consistent separation performance, and reduced labor requirements. The amount of the mixture used for the separation has no limits as for batch distillation. Following are some terms that you need to know for better understanding of the diagrams. The feed is basically the liquid that is fed into the rectification or the distillation column. The tray below the inlet nozzle, as you can see in the red lines, is called the feed tray. Heavy components are the component with lower relative volatility and higher molecular weight. So they are majorly found in higher concentration in the bottom part of the column. Whereas light components are components with a high relative volatility and lower molecular weight, and they are majorly found in higher concentration at the top part of the column. The products which leave the top of the column are termed as top products. This product is usually passed through a condenser, liquefied and stored in a receiver, as you can see here. The bottom product is the product that leaves through the bottom of the column. A reflux is a portion of vapor from the top of the column which has been condensed to a liquid and returned to the column as a liquid from the above top tray. And then reboiler is a heat exchanger at the bottom of the column which boils some of the liquid leaving the column. The vapor generated returns to the column at the top of the stripping section. So to summarize the working of the continuous distillation, the feed which is in liquid enters the distillation column, where the components will get separated based on their relative volatility. Heavy components will be found at the bottom part of the column, whereas light components will be found in the upper half of the column. The vapors containing different components will be passed to the condenser, which will convert them into liquid back. Some part of the condensed liquid will be sent into the receivers, whereas some part will be sent as flux back into the distillation. This is done to make sure to separate components which have similar volatility. The last method to be discussed to carry out the concentration in the downstream processing is the supercritical fluid extraction. It is a process of separating one component from another using supercritical fluids as the extracting solvents. A supercritical fluid is any substance at a temperature and pressure above its critical point. Critical point is a point beyond which the liquid and its vapor do not exist as distinct or separate liquid and gas phase. Rather, the liquid and its vapor can coexist. It can effuse through solids like a gas and dissolve materials like a liquid. Now this is important. The solvents that can be used as supercritical fluids. The first and the most important one is carbon dioxide. It is preferred because it is non-toxic and non-hazardous. The other solvents that can also be used are nitrous oxide, sulfur dioxide, ammonia, ethane, propane, butane, pentane and ethylene. The next slide shows some important points that how the supercritical fluid extraction method takes place. Supercritical fluids, when above their critical temperature and pressure, exhibit unique properties that make them excellent solvent, as we have discussed in previous slide. They have a density similar to a liquid and a diffusivity similar to a gas. By adjusting the temperature and pressure, the solvating power of the supercritical fluids can be tuned to selectively extract the target compounds of interest while leaving undesirable components behind. One of the main advantages of this method is that it can be performed under mild conditions compared to other extraction methods. The process is typically carried out at temperatures below those that can cause degradation or denaturation of biological products. This helps in preventing the integrity and functionality of the target components. Further, supercritical fluids such as carbon dioxide have good solubility for a wide range of organic compounds. By adjusting the pressure and temperature, the solubility of the target compounds can be enhanced, allowing for efficient extraction. This method is often considered as environmentally friendly extraction method. Supercritical carbon dioxide is non-toxic, non-flammable and readily available. Additionally, it does not leave behind harmful residues or solvents in the extracted product, making it suitable for applications in the food, pharmaceutical and cosmetic industries. After the extraction is complete, the supercritical fluid can easily be separated from the extracted compounds by simply reducing the pressure. Now this is important. The supercritical fluid then returns to its gaseous state, leaving behind a concentrated extract of the target compounds. Now this concentrated extract obtained from supercritical fluid extraction can undergo additional processing steps to purify or further refine the target compounds. As you can see in the diagram, the supercritical fluid extraction unit contains two columns. The first is the extractor unit, that is the unit for extraction. The second one is the separator still, which is used for separation of the product obtained from solvent using carbon dioxide. The solvent that we use here is a supercritical fluid at a high pressure of about 72 bars and a temperature of 31 degrees Celsius. The fermentation broth is pumped into the extraction unit and contacted with the solvent which is entering from below. The two streams obtained are the extract phase, which is solvent rich, and the raffinate, which is solvent deficient. Raffinate is withdrawn from the bottom while the extract phase is brought to a lower pressure in a separator still, where the separation between the product and the solvent is achieved. The solvent is later compressed to high pressures by means of a compressor pump and recycled back into the extractor unit. And finally, the product is withdrawn from the separator still. So with this, we end up with the second stage of downstream processing, that is concentration. In next session, we'll discuss stage 3, that is purification methods, which includes different type of chromatography like adsorption, ion exchange, gel permeation, affinity, high performance, and liquid-liquid chromatography. Membrane processes such as reverse osmosis and ultrafiltration, liquid membranes, and crystallization.